Biosensor. So, a biosensor is a self contained integrated device that is capable of providing specific quantitative or semi quantitative analytical information using a biological recognition element which is in direct spatial contact with a transduction element, according to IUPAC 1996. So, biosensors are used to analyze either it is quantitative or semi-quantitative, and biosensors are using the biological recognition elements like the enzyme, protein, etc. So, biosensor is a bioanalytical system, and an enzyme electrode is a biosensor. So, enzyme electrode is the example of biosensor. Next, biosensor. Any device that use specific biochemical reaction to detect chemical compound in a biological sample. So the blue laser light is pass on, and the yeast cell having the DNA gene, and also it is giving the specific reaction with a particular cell and chemicals used in the detection process. So ultimately biosensor is used for this purpose. Now current definition: a sensor that integrates a biological element with a physiochemical transducer to produce an electronic signal proportional to a single analyte, which is then conveyed to a detector. So ultimately biosensors are having the biological element, and they having or they can bind with the transducer, and ultimately from that electronic signals we are getting the results. This is the simple mechanism of biosensor. There are so many biosensor used, like enzyme act as a biosensor, antibody act as a biosensor, microorganism act as a biosensor, and so many. So analyte, it means the sample we want to detect, so it is named as analyte. Analyte bind with the bioreceptor. So here the bioreceptors are enzyme, antibody, and microorganism. Then next they are transferred to the transducer. This binding is transferred to the transducer. So in transducer we are using electrode, photon counter, piezoelectric device, or semiconductor. We are using it as a transducer. Now they measure the signals. So via the measuring signal and electrical signal, the detector showing the result. The father of biosensor is Professor Leland C. Clark in 1980s to 2005. History of biosensor, it is not that much important, but now basic characteristic of biosensor is linearity, sensitivity, and selectivity, and response time. So we are detecting the substrate in the linear concentration. The value of electrode in the response to the substrate concentration is a sensitivity of that particular electrode. Now selectivity of the chemicals is also affecting the result, and response time. So this is the simple mechanism or method to describe the biosensor. First one is the sampling, handling, and preparation of samples. We can say analyte. Now detection process is done via the enzyme, antibodies, and etc. So many. Now this analyte and particular biosensor bind with each other and give the signals. That signal will analyze into the transducer, and due to that electrical field generation, we are getting the response from that process. Biosensor required first the analyte. Analyte is what do you want to detect. So we can detect the protein, toxin, peptide, vitamin, sugar, metal ion, etc. Now sample handling. How to deliver the analyte to the sensitive region? So by the microfluid disc, concentration increase or decrease, filtration and selection process is done when we are handling the sample. Next step is detection and recognition. So the analyte are detected and recognized by the transducer. Next is signal. So after detection, the signal will generated. Example of biosensor is pregnancy test. So it detects the HCG protein in urine of females. Next is diabetes patient using the glucose monitoring device and monitor the glucose level. Other example is the infectious disease biosensor from the RBS. All the time coal miners biosensor are there. Typical sensing technique for biosensor. So typical detecting method like fluorescence, DNA microarray, SPR surface plasmoresonances. These are the techniques which are used to detect the biosensor. Type of biosensor are the calorimetric, potentiometric. In potentiometric we are using the electrodes, calorimetrics. Next is optical biosensor is piezoelectric biosensor. Piezo biosensor is device use the gold to detect the specific angle at which electron waves are emitted when the substance is exposed to the laser light, such as a quartz which vibrate under the effect of electrical field. So in this the gold is used to detect, and the vibration via the electrical field is used to detect. Electrochemical biosensor, so the movement of electron in the reaction process is detected between the two electrodes. Potentiometric, for the voltage change, the distribution of the charge is detected by this. And next is optical biosensor. So via the calorimetric, for the color, the measure the change in the light absorption. Calorimetry is the method to measure the change in light which is absorbed. So optical biosensor need the calorimetric. Next is photometric for the light intensity. Via using photometric method, we are finding out the intensity of the light. Calorimetry enzyme reaction is exothermic, so it is used to measure the difference between the reactant and product and analyze the concentration. Electrochemical DNA biosensor. So the DNA hybridization biosensor are there, and formation of DNA recognition layer. Ultimately hybridized event is done and transformation of that hybridized into the electrical signals. DNA biosensor. So application of this kind of biosensor into the diagnosis process and mutation process. So type of DNA biosensor are the electrodes, chip and crystals. Wearable biosensors. So the smart shirt and ring sensor are the wearable biosensor. At the nanoscale, it is also used. Application of biosensor. So in your syllabus, the application and general principle of biosensor is mentioned. So food and analysis. For the food analysis, biosensor are used. Study of the biomolecule and their interaction. To study the biomolecules which are getting from the biological origin. Drug development, crime detection. Also biosensor are used in the crime detection. For example, fluorescence is used to detect the crime in forensic. Medical diagnosis, both at the clinical level and laboratory level. Environmental field monitoring, by using biosensor we are monitoring the environment, as we can say it is now quality control, industrial process control, detection system for biological warfare agents. Next is manufacturing of pharmaceutical and replacement organ. So in pharma industries, for the manufacture purpose, biosensor is used, and when we are replacing the organs in the human body, the biosensor also used. Now this play a part in the field of environment quality, medicine and industrial, mainly by identifying the material and the degree of concentration present. So here, by using biosensor, it is useful in the environment quality. Next, it used in the medicine and industries, in which we are identifying the material and concentration of that material. Thank you.